Hello everyone, today we're gonna focus on the cloning method, which is circular polymerase extension cloning method for combinatorial DNA libraries, complex cloning. We focus on this article, and as an abstract, it is our article that we focus on the circular polymerase extension cloning method, which is beneficial to use for complex and combinatorial DNA libraries. This method is based on polymerase overlap extension and free for restriction, digestion, ligation, like that. And exactly, it is the efficient, simple, and economic method we will mention in details in next slides. Again, I can clearly say that it is circular DNA assembly and cloning method, which is simple, inexpensive, efficient. We can generally use it in proteomics, genomics, and synthetic biology. It is special cloning method is that that is sequence independent and ligase free. That generally use the polymerase extension mechanism to join overlapping DNA fragments into a double-stranded circular form. And in the typical reaction, we can clearly say that the linear double-stranded insert and vector firstly heat denaturated. Single strands be gained, exactly. Then anneal with the overlapping ends and we gain double-stranded circular plasmids in that line. That is an important point is that all overlapping regions between insert and vector are unique and carefully designed. Because they are carefully designed that insert vector have very similar and high melting temperatures that gives that is the factor gives elimination vector re-annealing and concatenation of insert, and that makes our cloning method more efficient. We can clearly say that. And CPEC cloning has its root polymerase cycling assembly. CPEC procedure not only single gene cloning, but also high efficiency library cloning, multi-way cloning, and combinatorial library cloning. It is the subject of our article, we can clearly say that. CPEC is different from the polymerase cycling assembly in that our cloning method forms abundant circular products of distinct lengths, but PCA, which is polymerase cycling assembly, forms a smear of linear products. In the correct size products can only obtained by further PCR amplification and PCA products. We can clearly say that. And there is some existing sequencing independent cloning methods that including ligase independent cloning, sequence and ligase independent cloning, uracil DNA, glycolysase cloning, and clone tank infusion cloning. And I want to add something in here, including ligase independent cloning and uracil DNA glycolase cloning require the presence or absence of certain types of nucleotides at specified location in overlapping regions, making these techniques less sequence independent. It is the additional knowledge for that. Again, CPEC cloning method is efficient, simple, speed, economic. However, there is some intrinsic limitations that are DNA polymerase and overlap extension method that some limitations in size of plasmids and total number of fragments, what we exactly say. And also, it is schematic diagram, as you see, of circular polymerase extension cloning of combinatorial gene libraries, as you see in the figure. As you see in the figure, two gene libraries cloned in a frame into a vector. Vector inserts share overlapping regions at ends. After denaturation, as you see, an annealing process, inserts and vector extend to use each other to complete full of circle. Assembled plasmids are ready to use for transformation. We will talk about in the other types of the slides. Let me show you experimental design outline. Design of overlapping sequencing, vector and insert, as you see, preparation of linear vector, preparation of inserts, CPEC cloning, and transformation. It is the experimental design outline. It is the primer design of the R cloning type. As you see, is in A, an example, a single insert cloning. It is an example. The primer is designed to amplify the gene insert, introduce overlapping regions with the vector. In the B, we use our specific gene, LAX-Z alpha gene, library cloning. We will talk about in the results clearly. And the C, overlapping regions in combinatorial library cloning, as described, again, in result we will talk about, as you see in the primer designs in the part of C. First of all, we begin with insert preparation. Set the PCR on ice, which showed in the table. We set the PCR in that way. When the amount of the library DNA increase, template should larger than standard PCR. It's the point. And run the PCR like in the table, as you see in the figure. And finished PCR product, PCR reaction, combined with DNA loading dye and run on the agarose gel. And unique bonds we will gain. Unique bonds are at the desired position. We cut it with sharp razor and measure its concentration. That's the point, what I can say. In the second part of the preparation is the linear vector. Again, set the PCR as you see in the table and run the PCR with the following conditions as you see again. Again, finished PCR combined with DNA loading dye and run on the agarose gel and unique bond that we expected, cut and measure its concentration is the same with the previous slide. And CPEC of single or combinatorial gene libraries is this, the other part. In the other part is that cloning reaction is prepared, as we see in the table, as you see again, and insert and vector set molar ratio generally set between 1:1 and 2:1, as you see, it's writing in here. And then the other part is coming that. And run the cloning solution with the following conditions, as you see the condition in the figure in the table, and we will talk the number of cycles that decided according to complexity of the cloning in the results. I can clearly say that we will talk about it, and finish the reaction combined with DNA loading dye again and run at the agarose gel and visualize the result product in under the UV. We can clearly say that, and we know that exactly. It is the transformation part of colony reaction. We aim to pick desired number of isolated colonies, exactly, and we will talk detailedly in the results in the next slides. We can talk in it. And then there is the optional parts that aim to understand picked colonies have insert in correct size. Colonies picked directly from the plate, set the PCR, run the PCR, and observe in UV to determine presence of inserts. And important thing is that their size, understanding the determining their size is important in that part. It is troubleshooting part. There can be some problems in the experiment in the cloning part, like many colonies yield PCR products of a wrong size. Maybe it can be a problem. That is the possible reason is that did not use high quality library DNA that can be fixed by solution, and maybe did not select the colonies with positive reporter gene signals. Is the important point is that again can be a possible reason. And another problem, little or no PCR products we gain. And possible reason is that did not use appropriate DNA polymerase for the colony PCR. We can fix it to solve this problem. That is a troubleshooting that all experiments have. Exactly, there are some problems, possible reasons, and we can solve these problems by following these things and like that. And it is results for our experiment is that it is circular polymerase extension cloning of LAC-Z alpha gene library. In the first figure A, as you see, in fourth lane, as you see in here, we see the cloning products that upper bands full length of the cloning products. In the lower part, the other one is empty vector, as we see. Let me mention the B part. In B, E. coli colonies expressed small portion of library LAC-Z alpha gene codon variants. We see clear blue color on the plate, as you see, when we compare with the C. What the C with C is represent control parts, represents E. coli colonies again expressed wild type Z alpha that we observe blue color, but not as clear as like in figure B. We can compare with this, and then also results according to cycles. In that results that examined the cloning of fusion libraries between codon variants of LAC-Z alpha and GFP genes. The results show that clearly higher level of cycles is more useful, more beneficial for combinatorial libraries. As you see, when we compare the cycle number 5, 10, and 20 terminal cycles. As you see, lane 2, 3, and 4, we increase the gain, and we can clearly say that a higher level of cycles is more useful for complex cloning or combinatorial libraries, DNA libraries, we can clearly say that, right? And to sum up all of the article, all of the things, for the cloning method is circular polymerase extension cloning, is efficient, simple, economic, and useful for complex cloning and combinatorial DNA libraries. And it is beneficial method to use in genomics, proteomics, and synthetic biology. It is very beneficial method to use a lot of research, and it get be more common, and I like this type of cloning, which is simple and economic and efficient. And thanks for listening to me, and thank you.